was like, wait a second. In my pocket. Why did the football coach go to the bank? Why did the football coach go to the bank? To get his quarterback. Well done. I don't know what his quarterback was doing there. What do you call a really big ant? What do you call a really big ant? A giant. Nicely done. What did the dolphin shout when he got stuck in the seaweed? What did the dolphin shout when he got stuck in the seaweed? Kelp. Yep, that's right. All right, our last segment, vision anatomy of special senses. I think it goes without saying that we are fairly visual creatures. All right, we have two eyes. We have binocular vision. You know, our eyes are located towards the anterior aspect of our ventral surface. What do I mean by that? Well, they're not really out on the side here, like with fish, right? And so the significance of that is predatorial. Right? We're a dominant species. We can see what's in front of us. We have binocular vision. We have, when we're young, we have great close vision. And we have great far vision. And we can focus at different levels. We have some peripheral vision, but it's not as important for us to be paying attention to what might be sneaking up on us because we're probably sneaking up on something. Look at our cultures around the world. There's different types of foods that we've talked about with different smells and tastes, but also different appearances. So I have a student in the lab, an international exchange student from France, and their graduation is this Friday. And I'm really excited because as part of the international ceremony, because they're exchange students, they can't really be in the regular ceremony because they're just doing a semester and then they're getting their degree confirmed from their home institution. But NAU, I think it's kind of cool. They do a ceremony and we get to go. But the best part is they have international ethnic foods. And I'm excited to see what that banquet table is going to look like. It's going to be very colorful. Think about some of your favorite foods from your favorite ethnic background. There's a lot of visual importance to it. Look at fashion around the world. Look at the media. Look at internet ads. I mean, we are very visual. So this is another example of a sense, a special sense, where we interact with our surroundings, our outside environment. And we take this in all day long until we close our eyes. And we wake up the next morning, and it starts all over again. So, some of the external anatomy. I know it's been about 2 weeks now since lab, but this is the last stuff that you had. So it should feel like it tracks pretty well. We have structures, that's very familiar, like eyebrows and eyelashes. We know that these help with debris as well as sun interference, filtering the light, filtering the debris as it comes into our eyes. The palpebrae, which are the eyelids, we have an upper and a lower palpebrae. They actually come together at these lateral and medial commissures. That's where the eyelids touch on the medial aspect as well as the lateral aspect, these commissures. It's also where your morning sleep or your gunk sits, right? When you wake up, you have these little eye boogers. That gets stuck in the lateral as well as the medial commissure. The conjunctiva, that's the white of the eyes. Conjunctiva, the white of the eyes, it covers the eye lids and the outside of the eyeball. So your conjunctiva is on the underside of the eyelid. It's a mucous membrane that prevents or is keeping the external surface of the eye lubricated and moist so it doesn't dry out. And if you get a dry eye, then you get redness in the eye, and the whites of the eye or the conjunctiva looks a little red. The lacrimal apparatus, that's made up of the lacrimal gland as well as the ducts. So that's located on the lateral aspect, just above the eye. And every time you blink, a tear comes through the lacrimal ducts, runs across the eye, and drains on the medial aspect to this lacrimal punctum, drains into the lacrimal canal, and down the nasolacrimal duct into the nose. So, if you have a lot of tears that are flowing, inevitably you have a runny nose because the tears make their way down to the nasal cavity. Flushes debris, keeps it moist, filled with enzymes. This is an external surface of the body, so it's really important to flush this out. The next time you are looking in the mirror, if you kind of deflect the medial commissure, you can see this lacrimal puncture. It looks like a tiny needle hole. You probably never even noticed it. But you can actually see your own in the bathroom mirror. So, external anatomy. Pretty detailed. Now, let's talk about the internal anatomy. This is a slice that would be sort of a mid-sagittal view down the eye. So it's a sagittal slice, mid-sagittal view of the eye, not mid-sagittal of the body, for obvious reasons. You can see the conjunctiva that's found here, and you can hopefully kind of appreciate how it's on the inner side of the eyelid as well as the covering of the eyeball. In lab, you talked a lot about these muscles. We're not going to do that. We're going to talk more about the optical apparatus as well as the neural apparatus. How does the light come in? What do we do with it? And then from an action potential standpoint, how do we actually see? That's what we're focused on. But some of this anatomy that's review is, I think, kind of important to get us organized. So the eye is made up of tunics. The inside of the eye is made up of tunics. And tunics is a word that means coat. So there's 3 coats or layers. We're going to discuss those, and we're going to focus on the optical apparatus as well as this neural apparatus that I talked about. So first up, tunica or coat of the fibrosa. So this is the outermost layer or coat. And the sclera is really that white of the eye, that conjunctiva, but it's the white part of the eye that surrounds all the way to the back of the eye. And that coat is the conjunctiva plus layers that are deep to it, making up what we refer to as the sclera. So the conjunctiva would be the epithelium or the superficial-most layer of the sclera. It's made up of a dense, collagenous connective tissue. It's pretty tough. It's irregular in appearance, so it's strong in all directions. That should make sense. And then the cornea. And the cornea is a clear portion. Not the lens, but the clear portion that you actually place your contact lens on, if you have contacts. So that makes up the tunica fibrosa, the sclera and the cornea. The tunica vasculosa, also known as the uvea, is the choroid, the ciliary body, and the iris. The choroid has lots of blood vessels. It's pigmented, and it's located behind the retina, the choroid. So it's towards the back if you look in this slice. Choroid, behind the retina. The ciliary body. This is a muscular ring that surrounds the lens. So it controls what happens to the lens. And then the iris. This is the adjustable diaphragm, like a diaphragm in a camera. An opening, the iris, controls the size of that opening, or the pupil. The iris is the colored portion of the eye. So the iris is our tunica vasculosa, as part of the middle layer, if you will. We've got a couple of different colored eyes in this room. So if you have brown or black eyes, what do you think the pigment is? Dark colored eyes of the iris. Where have you seen that before? Melanin. And that pigmentation in your eyes gives the brown or the black. Now, if you have a lack of pigmentation of the iris, you're going to get what's called Rayleigh scattering, which is kind of why, is the sky blue? Is it really blue in color? You're like, well, is this a trick question? I go outside, it looks blue to me. I don't know. Well, it's really not colored. It doesn't have a color to it, but it is this scattering of light that comes off of a bulk of the coloration of the earth, which is water, so it looks blue. So the Rayleigh scattering phenomenon is scattering pigmentation from the back of the eye and gives an appearance of blue or green or hazel eyes. So the iris itself has the lack of color in blue, green, and hazel eyes. All part of the iris. Okay, our tunica interna, the retina. This is at the back of the eye. The retina is the structure that's going to connect to the optic nerve, where the image is going to be focused so that you can actually stimulate cones and rods to give you a picture. And we'll talk about the cones and the rods at the very end. Okay, so this optical apparatus, optics, like cameras, like what you have on your phone. There is an optical zoom, and then there's a digital zoom, right? The optical apparatus is what you would use in laboratory with the microscopes to see the glass slides. Optical apparatus is what you would use to see the stars, like a telescope. Optics, using different pathways of light through refraction and focusing of the image through lenses to give you a magnified view or a focused view. That's what optics is all about. So the cornea is the outermost portion of the optical apparatus and does a lot of refraction of light, the cornea. It also happens to be one of the largest transplants that happens. The largest number of transplants are corneal transplants. It's actually quite easy to do corneal transplants. Number one, it's avascular, so there's no blood vessels in it. It takes very well from one patient to another because there's really no blood vessels and a lack of significant immune cells. So corneal transplants, because your cornea can become cloudy as you age. And we'll talk about the cloudiness of the cornea and the lens here in a little bit. But the cornea is this outer portion of the optical apparatus. And then in the front here, we've got what we call this aqueous humor, which is in the front of the lens. And in the back, we have behind the lens, posterior lens, is what we call the vitreous humor, or the vitreous body. The vitreous body, you probably remember from your sheep eyeball dissection. When you poked into it, it kind of had a gelatin-like feel to it. So it's more gelatin, squishy in the back, and it's more fluid in the front that comes from the ciliary process, these arrows. It moves through the opening, the pupil, and then drains through the venous sinus. And the reason for these fluids, the back chamber and the posterior chamber and the anterior chamber, is to keep the eyeball inflated. And the reason you want to keep the eyeball inflated is because you want the light to pass through, and it needs to focus on the screen, like what's happening here. It needs to focus on the screen, and the screen has to be in the right position, because if it's too far forward, it's blurry. If it's too far back, it's blurry. All of you have probably played with, portable projectors, right? For an event, a party, a dance or something. You set it up in the middle of the room, you put your screen, you're like, it's blurry. You either adjust, right? You adjust the focus, or you move it back, or you move it forward, and you make it focus on the screen. So it's important for the eyeball to be inflated, and it's inflated with this fluid, not with air. So in the back, it's more of a gelatin-like or a jello-like substance, and in the front, it's more fluid. This scleral venous sinus that it dumps into is also referred to as the canal of Schlemm. That's a fun word, I think. The canal of Schlemm. That was probably the guy that discovered it. We're moving away from those terms, so we refer to it now as this scleral venous sinus. But what does this resemble to you? What does this fluid flow resemble to you? Where have you seen something like this, where you make fluid, it circulates, it drains? Where have you seen that before? Think. I like that. That was good. Any ideas? Cerebral spinal fluid. Now, in cerebral spinal fluid, do we ever have a situation that it would build up and have too much pressure? Right? Remember that? Hydrocephalus. So in this situation, if you have too much pressure building up here, it's glaucoma. And it's a similar type of procedure for glaucoma. You either use drops to lower the pressure, or you can actually implant shunts to drain that, just like what we saw in cerebral spinal fluid. Okay, the lens. The lens right here is connected via these suspensory ligaments, and when you get contraction or tension of the ligaments, it causes the lens to flatten. And when they relax, it causes the lens to open up. Okay, shifting gears to the neural apparatus. We'll kind of go back and forth now after we cover the neural apparatus. So the back of the eye, the retina, is really part of the brain. It's an outgrowth of the telencephalon. Remember, the telencephalon is part of the forebrain and the cerebrum. So it's technically, the retina is considered part of the brain. So you're actually focusing the light on a part of your brain. What you see, it's like, oh my gosh, I never unsee that. It's permanently burned into my brain. That's actually true. Right? The image that you see is forecasted onto your brain. I feel like that's so important to us. The sense of smell, right? Those neurons are the only ones that are exposed to the outside environment. That's pretty amazing. And here, this is the only place where you actually focus something, a picture, on your brain. I can't believe that is true. So this vitreous body, the gelatin, pushes the retina up against the posterior aspect, keeping it inflated. And on the back of the retina, we've got this area known as the macula lutea. And this macula lutea is this region kind of in the center. And as part of it, you have two structures. One is the center area of focus, known as the fovea centralis. In Latin, that means the central area of focus. That's what that literally translates to, fovea centralis. So this fovea centralis there's a high density of cones. Cones are for color. And visual acuity. It's easy to remember. Cones, C, for color, C. Not just color, but high visual acuity. So when you can see color, you have the ability to see defined shapes and features. So cones are not just for color, they're also for high visual acuity. And there's a high concentration of those cones in the fovea centralis. The optic disc is the location where the nerve enters the back of the eye, or the retina turns into the optic nerve and exits out the optic disc. The optic nerve, cranial nerve what? You remember? What cranial nerve is the optic nerve? Somebody look it up. What is it? 2. What's 1? He just put his book away. Olfactory nerve, that's right. Cranial nerve 2 is the optic nerve. Cranial nerve 1 is the olfactory nerve, which we already covered. Okay, so this demonstration won't work on this screen, but it'll work on your screen. So if you're watching the video at home later, you can do this. What I want you to do is where that optic disc sits and the optic nerve comes in, it's considered your blind spot because there aren't any rods or cones in that spot because that's where the nerve actually connects to the back of the eye. So you can kind of discover where that location is by doing this little trick. So you want to, you see a dot, a red dot or a red circle, and then an X on the screen. So if you cover your right eye and then look at the X with your left eye, and then move the screen or move your head closer or further away from the screen, and at some point it's going to disappear. And that spot at where it disappears is because that red image is trying to focus on the left eye right at your blind spot. Make sense? So let's say it again. Cover your right eye. Keep your left eye open. Look at the X, but out of your peripheral vision, you're seeing the red dot, and then you move the screen closer or further away from your eye, and at some point it's just going to disappear because that image is now being projected on your optic disc, your blind spot. Okay? Kind of cool. Okay, so the pupil itself, right? The pupil, how does it dilate? When we did this pupil dilation exercise, right? We talked about dual antagonistic innervation of the pupil in the peripheral nervous system lecture. So we've got a couple of different muscles here. We've got pupillary constrictors, pupillary dilators. So the constrictor muscles are circular smooth muscle. The dilator muscles are radiating out like spokes. And when they contract, they will adjust the size of the pupils. And this happens when you have a change in light intensity. So obviously in low light, you want the pupil to be wide open to allow more light to come in. And in bright light, you will constrict the pupils to prevent light from coming in. And this, you're pretty sensitive to this. Because, again, this image is projecting right on a portion of your brain. And so you're going to be very, very sensitive to this activity. So the lens itself. So the light comes in the cornea. It's going to pass through the pupil, and then it's going to pass through the lens. Now, the lens is going to adjust the image. It's going to fine-tune the image so that the focal point is right on the back of the retina. And that focal point right here, the image needs to be in focus when it's in this plane. And I'll show you here in a second, if it's in focus here, that's a problem clinically. If it's in focus back here, that's a problem clinically. But right now, what I want to focus on is the job of the cornea. So the cornea and the lens are both made out of proteins. And proteins, when they're exposed to ultraviolet light or radiation, over a long period of time, what do you think happens to those proteins? They denature. Great. That was Unit 1. So you remember the picture I showed in Unit 1 of a fried egg on the screen? I mean, again, you're not going to have pictures from Unit 1 on the screen on Monday. You're going to have only pictures from this unit. And we're not talking about denaturing proteins on Monday, okay? I'm just trying to connect some of the dots that we talked about like 4 months ago at the beginning of the semester. So when you crack an egg, that albumin, the white of the egg, is what color? Clear. And your proteins of your cornea and your lens are what color? Clear, because you need light to pass through them. What would make them go cloudy, like when you denature albumin in an egg and it turns actually white? Ultraviolet light. In the Southwest, we have a lot of sunlight. When we denature the proteins in the lens or in the cornea, we get aberrations called what? Cataracts if it's the lens. And that's what those cataracts, or in the cornea, you get aberrations in the cornea. Same thing happens in plastic. You can actually chemically denature plastic. How many of you drive a vehicle that's over 10 years old? Yeah, usually in a college classroom, it's, I actually drive a truck that's over 10 years old. When you bought your vehicle, were the headlights nice and clear? Now are they cloudy? They have cataracts. It's the same thing that happens in elderly eyes. Kind of weird, huh? So, refraction. The cornea does a tremendous amount of refraction. That's the bending of light. Because it's going from a refractive index of air into a clear protein that has a different refractive index. And so what happens is the light bends. As it bends inward to try to focus the image, and then the lens does a little bit more of the fine-tuning adjustment so that image is nice and clear on the retina. So an example of refraction. How many of you grew up with a swimming pool? How many of you ever helped your dad or your mom brush the pool and you put the pool brush in and it bent? The water's clear, you put it in. Or how many of you fish or spearfish? That's kind of a new fun sport these days. So if you see the fish, it's actually not exactly where you see it, right? Because light bends from air into water. And that bending of that pool pole as you're brushing the side of the plaster on the pool, and you see it kind of bend, or you put your hand in and your hand looks like it's broken, your arm looks like it's broken, because the light is bending from a refractive index of air to water. So if air is 1, refractive index, n equals to 1, the lens is 1.4, you can see the cornea is 1.38, and the aqueous humor, if you had air in there of 1, you would bend the light really weird. But if you put fluid in there of aqueous humor that has a refractive index that approximates the lens in the cornea, now the image stays nice and focused and in the right refractive range as it's making its way to the retina. That's why it's not full of air. Because I get questions like, why don't you just have the eyeballs inflated with air instead of with fluid? The other thing that would be problematic about air is compressible. So when you traveled up and down from Phoenix to Flagstaff, and we talked about the eustachian tube, and you're doing your right? Your eyeballs would be like, whoa, whoa, whoa. But now that it's fluid is non-compressible. Make sense? So this is actually a pretty good design. You follow all that? So now this light bending, it looks something like this. All right, you can see on this image here, normal on the lower left, you can see where the focal plane is, the refraction of the cornea, the fine-tuning of the lens, the image is projected on the retina. This image nicely characterizes where cranial nerve 2 comes in, and at this location, that is the blind spot or the optic disc. But the image is focusing up here. Now, if you've got a situation known as hyperopia, farsightedness, you actually can see things far away, but you have trouble on focusing things that are close up. Farsightedness, hyperopia. You can't see things close up because the image is actually focused beyond the retina. So it's blurry right here. So what you do is you put a convex lens in front of it, and that adjusts the fine-tuning of the image so that it's actually corrected for farsightedness with a convex lens so that the image is in focus right here at the retina. If you're dealing with myopia or nearsightedness, that means that you can see things close up, but you can't see things far away. In that situation, you use a concave lens. It bends the light so that it focuses in the focal plane. So, emmetropia, normal. Hyperopia, farsightedness. Myopia, nearsightedness. And the farsightedness means you see things far, but you can't see near. Nearsightedness, you see things near, but you can't see things far. Make sense? It would be good to know these terms and designations that are associated with each of them. Okay, so what's happening with presbyopia? This is a loss of lens flexibility. This occurs as you get older. A loss of lens flexibility, which is one of the reasons as you get older you need these guys, reading glasses. Usually that happens about the age of early to mid-40s. Okay? In some patients it happens later. Like for me, I just haven't started using these last year. So, and I'm almost 50. So some patients it's like at 41 they're using reading glasses. But between 40 and 50, most everyone's kind of reaching for reading glasses. Presbyopia. Okay, the other one is astigmatisms. So astigmatisms are like an optical defect in the cornea. So now what happens is you've got adjustments that need to be made using corrective lenses to account for more aggressive type of focusing. Because remember, the lens just does the fine-tuning. So if you have astigmatism, you might have gone to the eye doctor and like, well, you have an astigmatism, so it's more complicated. You're like, okay, what's that mean? I'm special? That just means that one of your cornea on either eye. Have you ever been to like a carnival and you go into the House of Mirrors? There's like a normal mirror, and then there's one that makes you look all like squiggly, one that makes you like really wide, one makes you really tall. So there's aberrations in the mirror that are causing aberrations in the image, right? So that would be the example of astigmatism. So that's happening with your cornea with an astigmatism. So you have to have an astigmatism correction. Okay, so glaucoma. This is a group of eye diseases that can lead to blindness. And why could it lead to blindness? Why do you think that might be one of the outcomes in glaucoma? How can high pressure in the anterior chamber of fluid lead to blindness? What do you think's happening there? Use what you think you know. Yeah. Okay, that's a good theory. Does it block incoming light to the eye? It doesn't, because the fluid is still clear. So with glaucoma, you make too much and it builds up in pressure, but it's still clear in color. But it's a good guess, yeah. The pressure affects the optic nerve. How does the pressure affect the optic nerve? That's correct. You build up pressure in the anterior chamber, which puts pressure on the posterior chamber, which puts pressure on the retina, and that puts long-term pressure on the optic nerve, and that's the problem. That's why we monitor it very closely. So every time they say you should go to the eye doctor at least once a year for just a checkup, and as you get older, they will do tests for glaucoma for eye pressure. Okay, the last little segment that we have to cover, the vision physiology of how this all works. So these are photo-bleaching pigments in the eye, which is kind of amazing. You all are probably too young, most of you are too young, to remember projection televisions. Does anybody remember projection televisions? No? That's what I thought. Oh, somebody does. Okay. All right, sweet. Been to a museum lately? That's awesome. So projection televisions consisted of 3 colors: a red, a green, and a blue color. And from the red, the blue, and the green color, you could actually get all the primary colors and see color television. So this is basically what is built off of the eye and how we see color. We have what we call cones that are of that variety. They're of red, blue, and green. And what that means is when they are triggered by light, they send a signal to the brain to interpret the wavelength of light that they're seeing as being in the green wavelength, or in the red wavelength, or in the blue wavelength. So, to orient you, this is the front, and this is the back of the eye. The light's coming in. You see this little flashlight. The light's coming in. It came through the cornea, went through the pupil, it went through the lens, and it passes right by the nerve fibers, which is weird to me. This is very strange to me. It passes right by these. It passes right by these ganglion cells, passes by the bipolar cells. It actually goes all the way to the back of the eye where the image is focused. It goes right past all these. And as it focuses on this retina, you have these individual rods, are these guys. They look like rods. These guys have cones. They look like cones. When they actually sense the light as a photoreceptor, it sends an action potential back this way towards the front of the eye, which is weird to me, but to these bipolar cells and then to the ganglion cells, and ultimately the ganglion cells converge into nerve fibers that go to the optic nerve. So the light passes it bypasses all of this and focuses here on the screen, which is the retina, which is where your photoreceptors are embedded in the retina. So it's pigmented epithelium. And above it, that's what we're saying, above it are 3 layers of cells. But the main characters are these photoreceptor cells. Now, the cones number about 6.5 million in each eye. And the rods about 130 million in each eye. And they converge on a limited number of bipolar cells, like what we just showed. Right here, bipolar cells, the middle ones. And those are our first-order neurons. The second-order neurons are these ganglion cells. And the axons from those ganglion cells, you can see all these axons bundled together and actually form cranial nerve 2, which is the optic nerve. So this is all happening on the retina. And we have these visual pigments in the rods and the cones, and we have 2 different types. So the rods are more simplistic. They have rhodopsin, and rhodopsin's made up of an opsin protein and a retinol. And then in rods are for black and white vision, night vision. And the rods in your eyes are predominantly located more on the periphery. Remember, this fovea centralis in the middle is the highest concentration of cones for visual acuity. And on the periphery is where your night vision is located. So try this technique the next time you get up in the middle of the night in your dark bedroom. Or you're navigating through the kitchen to get a glass of water and the lights are off and you don't want to turn the lights on because then it blinds you. And we'll talk about that here in a second. Try to look at where you're walking out of your peripheral vision. And you'll actually have better visual acuity because you have more rods on the periphery. So if you're hiking at night, you're recreating at night, you're walking to the bathroom in the middle of the night, if you kind of look more at the periphery and you're walking this way, you'll actually have better visual acuity out of the periphery because that's where your rods are mostly concentrated for night vision, low light. You can also do this. It's a beautiful exercise in Flagstaff because we have wonderful skies. Go out at night on a clear night, look up at the stars, find a cluster or an area that you want to see, and then look a little bit away from it and watch, it'll get a little brighter. You'll see it kind of brighten up because you're lighting up more of your rods by looking on the periphery. Try it. It's kind of cool. So they have two rods have rhodopsin, and they have an opsin protein and a retinol protein. We'll talk about that here in a second. And you can see the configuration that's shown here, like in purple. Right? You can see specialized cell with a nucleus. It's packed with mitochondria, and it's got this outer segment, and it's in a rod shape. That's where it gets its name. You can see on the scanning electron micrograph the rods versus the cones. The cones are shorter in stature, and they got like a pointy tip, like a cone. The cones have photospin, and they have three types: red, blue, and green, just like we talked about. So the cycle that I'm going to show you is for night vision, for these rods, for the opsin and retinol, but it applies to the red, blue, and green for cones. So let's just walk through this. And it'll show you how it cycles in our eyes so that we can see things. Where are we? We're at the back of the retina. Light's coming in. It's activating these photoreceptors. And these photoreceptors are specialized to see certain wavelengths of light. Red, blue, and green. Or in this situation, light or no light. Like white, full spectrum light, or no light. So black or white. So let's start at the top. In the light, this is rhodopsin. So this is your rhodopsin molecule that's made up of the opsin and retinol. Opsin and retinol. This is called cisretinol. This is a chemistry term that talks about the three-dimensional architecture. So this is what we call in the cis orientation. Okay, so it's slightly bent. Now, when light hits it, the rhodopsin absorbs the photon of light, and this cis-retinol goes through a conformational change, or what we call isomerizes, to trans-retinol. You see how it straightens out versus being bent? And this opsin trigger, when it does this, when it moves from this format to this format, that triggers a cascade that breaks down cyclic AMP. That's a second messenger system inside of a cell. As that breaks down, this ceases what we call the dark current, and now you create signals in a light current that are perceived as an action potential. Doesn't stop there. The opsin is dissociated from opsin. The retinol is dissociated from the opsin. And then when the light goes away, these actually recombine in the dark cycle to form rhodopsin, which is your opsin and your cisretinol, to recycle this. This takes a little time for this to happen, a few seconds, which is why if you step out of a theater into bright light and you're blinded, and then you go back into the theater, you can't see because everything is photo bleached. Once these guys dissociate, you're basically in a refractory period from an action potential. So you go out into a bright light, you're blinded by light, you go back in, you can't see, it still looks bright. It still looks bright because the action potentials are still being sent even though you're in a dark room. So this photo bleaching dissociates the opsin from the retinol, And that triggers your action potential cycle, but it obviously renews, right? The photobleaching. Takes a little second, but it comes back. Questions? That's everything that I have for you on vision anatomy and vision physiology. And that concludes our lecture for the semester. The only thing remaining is your final exam on Monday, 3 o'clock in this room. But hold tight.